Hello everyone. So some people wanted a video about monoclonal antibodies, and this is going to be kind of a long video because monoclonal antibodies are complicated, and I think it's nice to have a little bit of historical context and background information to understand monoclonal antibodies. And I really like the history of medicine and the history of science because I think it's very interesting. So we'll go through a little bit of that. Antibodies, the history of antibodies kind of starts with this guy, and I'm probably going to butcher their names because I'm not German, but Emil von Behring and Paul Ehrlich. These two guys worked in the late 1800s, and what they figured out is that for diphtheria and tetanus, these are diseases that where the bacteria make a toxin, they noticed that people that had diphtheria or tetanus were not susceptible to getting sick again from it, and they figured there was something in their blood that made them uh, resist the toxins, and they called this antitoxin. And they did work in like animals, guinea pigs, and things like that, where they would inject them with diphtheria toxin and tetanus toxin. And they realized that if they took the blood of one animal that had been injected and they spun it down and just took out the serum of from that animal and put it into another animal, they could transfer this protection against the toxin, and they called it antitoxin. And back then, lots of children were dying from diphtheria. It was one of the most common reasons that children died. And they found an animal that was large enough that they could get a lot of blood from it. And they used horses actually, and they would basically inject diphtheria toxin into horses. The horses would make this antitoxin, and they would get the blood from the horses, and they would give it to children. Not the whole blood, but after they just the serum of the horse. So it didn't have any of the cellular components, so they wouldn't have get sick from it. Um, and an interesting story is when they were doing this, it was like there was a war going on in Prussia and Germany, and so all the horses that were good horses were being used in the war effort. So he got all the horses that were old and supposed to be slaughtered, but they were treated quite well because they were thought of as kind of heroes in saving children because they were being used to make this medicine for the kids. Paul Ehrlich actually helped him with this, and so for this work, Emil von Behring he just called it antitoxin. Later on, people figured out this was antibodies. He got the very first Nobel Prize in 1901 for that. Paul Ehrlich actually helped him with this, but he did not get the Nobel Prize in 1901 for it. He got it later in 1908 because he further did a bunch of work and figured out that these antitoxins or antibodies, as he would later call them, must be a branched thing because it would stick more than one thing together. So he figured it must have more than one active site where it's sticking to things, and he turned out to be correct. And he also realized that mothers could transfer this protection to their children, and they were wondering, is this a genetic thing where like they would genetically pass it on to their children? Because he did these work in mice, and he would make the mice resistant to ricin, which is a poison derived from castor beans. And they noticed that the mothers, their babies would be resistant to the ricin. If the mothers were resistant, the babies were too. And so they were trying to figure out if it came from the genes or if it came from some other method. And so Paul Ehrlich did a bunch of very ingenious and elegant experiments where he like basically he had fathers that were resistant but mothers that were not resistant, and then he found out the babies weren't resistant. So they definitely didn't get it from their fathers. And then he actually swapped the babies. Like he put the resistant mother's children on non-resistant mothers, and he found out that the babies soon became non-resistant. So after all these elegant experiments, he figured out that it was coming through their blood, I mean through their milk, that mothers were transferring their immunity to the babies through their milk. So that was another thing that he discovered. Anyway, this is where the idea of antibodies first came in, and then later on, these two guys, Gerald Edelman and Rodney Porter, they simultaneously figured out the structure of antibodies. And the structure of antibodies is basically like a Y. It's got like a stick, and it branches, and there's two active ends like that stick to the antigen. And these two guys figured it out, and you can actually read their original papers This is, I think this is Edelman's paper, dissociative gamma globulin, and then this is Porter's paper, hydrolysis of rabbit gamma globulin antibodies with crystalline pathway. Well, anyway, this is the paper that got him a Nobel Prize right here. Um, so they figured out by doing various experiments that the antibodies must be made of two subunits. And now we understand that this is what antibodies look like. There is a heavy chain, which is this long thing right here. So there's two heavy chains, and they're linked by two disulfide bridges. And then there's two light chains right here. And this area is called the fraction crystallizable, because you could crystallize this because it had a constant structure. And this was called the the uh, the fab portion, which is the part that sticks to the antigen, and you couldn't crystallize this part. And then later on, they also figured out that up to this area is constant, and this area, the ends, are variable. So they called this the variable region, and this the constant region. So this area stays the same across all antibodies, and this area right here, the ends, change depending on what the antibody binds to. And probably this video is not long enough to go into the details of how antibodies are formed and how there's selection and stuff. But basically, you are born with all these possible antibodies that you may never encounter an antigen form. So an antigen is the thing that the antibody sticks to. So like if this is the antigen, the antibody will stick to it right here. And there's another word that you'll see in papers called epitopes. And epitopes are basically different parts of the antigen that antibody will recognize. So like one antigen can have several epitopes, several different areas that the antibodies will stick to. All right, so what Ehrlich and von Behring were doing, where they were making polyclonal antibodies. So what they were doing is they inject the toxin or the poison, or in the cases that we're now interested in, the virus or cancer cells or cancer proteins, and the 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 animal will generate multiple antibodies to the same thing. So not just one, multiple. And the problem with that was that sometimes the antibodies are really good and sometimes they're like not so great. And then you're also giving them a lot of non-specific antibodies, antibodies that don't even have anything to do with the target that you're interested in. And then sometimes they would get antibodies that bind the antigen but don't actually stop it. So they're called so antibodies that bind something but don't actually stop it are called non-neutralizing antibodies, and antibodies that bind it and stop it from doing what the bad things that it does are called neutralizing antibodies. And so you would like to have consistency and always get neutralizing antibodies. But with this polyclonal method, which is a bunch of different kinds of antibodies, which is how we naturally work, you can't guarantee that transferring that is going to get you um, exactly the antibody that you want. There's just no consistency and there's no predictability. That is when the idea, someone thought, if we could find the one antibody that is the neutralizing antibody and just make a bunch of that, that would be like the best thing. Um, and these guys figured out a way to do it. And these guys were Kohler and Milstein, and these guys won the Nobel Prize for figuring out this. And they figured out how to make monoclonal antibodies. And this is the paper published in Nature in August 7th, 1975, that got them the Nobel Prize for discovering how to make monoclonal antibodies. And the way this works is there is a type of cancer called multiple myeloma, which is a cancer of plasma cells. And plasma cells are the differentiated, um, the terminally differentiated B cell. And B cells are what make antibodies. And what they figured out is you could take a B cell that makes the antibody that you want, and you can fuse it. And they used a virus that causes cells to fuse with each other, called the Sendai virus right here. and they basically got the cells to fuse together. And this, what it did is it made a hybrid cell. It was immortal, like a cancer cell, and they got that from the multiple myeloma, and it would make the antibody, and it got that from the B cell that they fused it together. And what you could do is now you had this cell that you could grow infinitely. You could grow as much as you wanted, and it would just make one kind of antibody. And that's why it's called a monoclonal antibody. It comes from one clone. Um, so what the important thing here is they say alternatively, non-producing variants of, so they use the multiple myeloma cell that actually makes some weird antibodies. So their cells made some weird antibodies as well as the antibody that they wanted. So they said alternatively, non-producing variants of myeloma cell lines could be used for fusion. That is what people used later on. They found myeloma cells that don't really make anything, and they fused it so that the only thing it was making was the antibody that you wanted. And they were very prescient when they said this, and they said the cells used in this study are of all of bulb C origin, so that's a type of mouse. Um, it is possible to hybridize antibody-producing cells from different origins. Such cells can be grown in vitro in massive cultures to provide a specific antibodies. Such cultures could be valuable for medical and industrial use. That was like the understatement of the century. I mean, monoclonal antibodies have revolutionized research and um, basically now is in medical use. When I was first in the lab, that was like 20 years ago. It's been like 20 years since I used monoclonal antibodies. We were talking about how, um, 20, 30 years ago, we were talking about how monoclonal antibodies could be such a great thing, but it seemed very far off. But recently, there have been lots of medical things. So here's kind of a timeline of um, therapeutic antibodies, monoclonal antibodies. And you'll notice that the names are all MAB. They end with MAB. So if you see any drug where the generic name ends in MAB, that means it's a monoclonal antibody. And within the MAB, there's different kinds of MABs, like MUMAB is a mouse monoclonal antibody, and ZMAB is a chimeric antibody, and ZUMAB is a humanized antibody. Anyways, you see here, right, 1975, that's when uh, Milstein and Kohler made their first monoclonal antibody. And the very first drug was a anti-rejection drug in 1986 called OKC3 or Muromomab. And basically, this was a drug given to kidney transplant patients to keep them from rejecting their transplanted kidneys. It basically was an immunosuppressant. It knocked down the immune system so they wouldn't reject the transplant. Now, so that was the first one because they used this kind of antibody. What they used is they developed it in a mouse because Milstein and Kohler used mice, and that's how they knew how to do it. And they would basically create a mouse antibody that recognized a human target. Now it worked. It worked well, but the problem is that it's a mouse antibody. So after a couple of doses, your body would recognize, like, hey, this is a mouse protein. It's not a human protein. I don't recognize this. This is foreign. And your immune system says, I need to get rid of anything that's foreign. So basically, your humans will make an antibody against the mouse antibody. And so after a while, it stops working because as soon as this gets infused into someone, their body's antibodies will kind of neutralize it and get rid of it. And so these mouse antibodies were not very useful for a long period of time because after several doses, people would become basically immune to the drug and they would clear it out. So in order to fix this problem, people then went to what's called a chimeric antibody. And what they did is they would get this variable part of this monoclonal antibody and they would cut that out using molecular biology techniques. They would cut the genetic code of that out and fuse it onto the human constant portion. So these things were about 60 to 70% human and about 30 to 40% mouse. And so this was less immunogenic, and but it still was immunogenic. And a couple of like in I work in head and neck cancer, and one of the drugs that we use is this drug called cetuximab or Erbitux, and this is a chimeric antibody. And so it's okay for things like cancer where you're not going to get dosed multiple times for the same thing, because eventually people will mount an immune response to this. Next, they move to humanized antibodies, where basically they um, they tried to make most of it human, but there were still a couple of mouse things because they developed these systems in mice. So these were like 90+% human, and they still weren't had some problems because we will still recognize these small mouse parts, and eventually. So this was really immunogenic. This was a little bit less. This is a little bit less, and then finally, more recently, we've made fully humanized um, monoclonal antibodies. And these are kind of the state of the art. This is what we're doing now. And there's a couple of ways that we're making these. There's basically three ways that we're making these fully humanized monoclonal antibodies. One way is people have made transgenic mice, and basically transgenic mice are mice where we have manipulated their genes so that we can they're like a strain of mice that their genes are completely controlled by us. And these mice were mice that had no immune system, and basically we genetically engineered a human B cell immune system into them. So they make human cells. They make human antibodies, even though they're a mouse, because we've put human antibody genes into them. So you can just immunize these mice, and they will process the antigen, and they will make human antibodies for it. And then you can take those cells out and stick them on a hybridoma. That's where you fuse it with a multiple myeloma cell. That's what Milstein and Kohler did in 1975. Those fused cells are called hybridoma cells. You make hybridoma cells, and you can make a fully human um, antibody that you developed in a mouse transgenic mouse system. The problem with that is that the antigen processing is still a mouse, and so they don't process the antigen exactly like a human does. So these antibodies are sometimes not quite as good. Another way they did it is this way, and this was so ingenious. These guys figured out a way of doing phage display, and what this is let's say that I was infected with a virus and I survived the virus. And now I have a bunch of antibodies against this virus. And people would like to use my blood to create a drug that fights this virus. So then I would donate some of my blood, they would get the B cells from it, but then how do you know which of these B cells have the antibody, because I'm making thousands and thousands of antibodies, which one is the one that they want? And there's one way to do it is to just check each one, but the other way to do it would be to do what's called a phage display, and you process all the cells, you chop it up, and you make a bunch of the genes for um, every single antibody that I'm making, and then you can check those. This is a very easy way to check those genes. So one way to check those genes is to put them in plasmids, which are little circular pieces of DNA, and have them express it, purify the protein, and check the protein. And you'd have to do that like hundreds of thousands of times, which is very labor intensive. What these guys figured out is there's a thing called a bacteriophage, which is a virus that only infects bacteria. And they figured out if you clone the genetic material into this virus, it will display the actual variable part of the antibody on the surface of the virus. So you basically have a little, think of it as like an envelope that has the antibody, and inside the envelope is the gene, and outside the envelope is the little antibody. And now you can just basically put the antigen or the virus that you want to fight off like onto a sheet, and then you can wash these phages over it, and all the phages that recognize the virus will stick to it, and all the phages that don't recognize the virus will not stick to it, and you can just wash them off. And all the phages that are stuck to it, those are the ones you want. And then you can wash those off and open the envelope, and then you've got your gene. So this is something called phage display, which is a very powerful technique for screening lots of genetic material, and these guys found it. Now, the last way of doing it um, is something called single B cell cloning. And this is like probably the most advanced way of doing it. And what they do is they isolate single B cells, and they basically use flow cytometry, which is this way of labeling an individual cell with a fluorescent tag, and then using like a laser to check their fluorescence, and if they glow they get sorted one way, if they don't glow they get sorted another way. And so what you do is you get plasma cells, and the reason that this is great is because this part right here. So class switched memory B cells and antibody secreting cells, which are plasma cells and plasmablasts, are of special interest to obtain monoclonal antibodies as they bear somatically mutated B cell antigen receptors with high affinities. So this is what they're talking about is something called somatic hypermutation, and maybe if you guys are interested, we can talk more about somatic hypermutation and the way our immune system works. But basically, there's a way that our body makes the antibodies even better. And if you wait until you get to these plasma cells, they've already done it for you. And you can screen them with this flow cytometry by trying to tag them with the virus that you want it to attack. And then by doing this, you can isolate the single B cell out of all the cells that are floating around your body that has the exact antibody that you want. And once you find that B cell, you can extract the DNA out of it and clone it into a plasmid, and then you can make a bunch of it by transfecting it into another cell. And you transfect it very similarly to these mRNA viruses, where you encapsulate the genetic material into basically a lipid nanoparticle of sorts, and have that merge with the target cell. So the target cell will now make the antibody that you want. Just like with the mRNA vaccines, they make the spike protein that you want. Um, okay, so that's essentially how monoclonal antibodies are made now. Either they're made with transgenic mice, or this phage display, or with this single B cell cloning. And in the next video, we are going to kind of go through the different monoclonal antibody therapies for COVID. There's several of them. The most common one we know about now is probably the one from Regeneron, which is 2 monoclonal antibodies. But we'll kind of go through each one of those and see how they were developed, and look at data on how well they work. So I hope this video gave you guys some idea of what monoclonal antibodies are, how they're made, and what the problems were initially, and how those problems have been solved over the years with various techniques. If Please comment below if you thought this was a little too long, too technical, maybe not technical enough. You want more detail, less detail, and I'll try to kind of tailor future videos to that.